Good morning, everyone. I'm welcome you all to the 16th day of CSIR Life Science Question and Answer discussion session. As per our schedule, day 15, day 16, and day 17, we are discussing the Unit 5 question, Part C questions, that is developmental biology portion. In this yesterday we have discussed some important questions. Today we are going to discuss the remaining questions. Just today I will go just fast because many of them having the personal routine works. Okay, we will directly start this session. Okay, first question is the capacitation of mammalian sperms allows them to activate it within the uterus and facilitate fertilizations. The following statements were made regarding event occurring during the capacitation. Here, the main thing in the question is this fertilization. In this fertilization, this process called the capacitation is the very important thing. In the process called capacitation, here the fertilizations occur, they are initiated in the process. Okay, it's or intermediate step in the maturation of mammalian spermatozoas. In this capacitation, penultimate step, it's a penalty step in the maturation of mammalian spermatozoas, and it's required to the fertilize the oocytes. And one more thing in the capacitation, here they mention activated in the uterus. They have provided the statement activated in the uterus and facilitate the fertilization. The following statements are made regarding the event occurring during the capacitation. What happened in the capacitation? As per their statement, correct one is C, PQR. So what happened in this capacitation process? Removal of cholesterol from the sperm head is the first step. Okay, removal of cholesterol from sperm head. Second one is removal of non-covalently bound glycoproteins. Okay, in this process, it's removal of non-covalently bound glycoproteins. This is the one. And increase the expression of fibronectin. If increase the expression of fibronectin, it influencing the attachment of the sperms into the oocytes. So these are all the process happen in the capacitations. Okay, next thing, forelimbs of human and flippers of whales. This is two things. Forelimbs of human and flippers of whales are embryologically homologous structure. Homologous means the similar structure. Why it's look like similar? Embryologically it's similar. What does the study of homology structure tell us about the evolution? Which is the correct one? Here PQRS is the correct one. I will explain you. So the forelimb of human and the flippers of whales are similar. Why? Because this is the example of adaptive radiation occurred due to the similar group of organisms habitating different environment. Whales are living in a different environmental conditions of their sea, in their sea level, sea heat. Likewise, humans also living in a various environmental conditions. This is the example for the divergent evaluation. Divergent evaluation means it's a combined character. Human having the various character. Okay, one human can live in Canada and also he live in Africa. So he have a capacity to live in various land. Divergent is a divergent character. They can eat anything, they can live, they were move through the water and can live in any environment. That's all the thing. And it's occur due to the organism habitating different environment and similar groups of organisms with the mutations is a very important process in this homologous structure. Why this mutation? As per their biological evaluation or evolutionary ancients, generations are comes from the reptiles and the water organisms. So this mutations, okay, this alterations happen in the water organisms that make a homological similar structure of the forelimb of human and flippers of the whales. Okay, it's occurred due to the variation getting the naturally selected different environment. These are all the three important reasons for the this revolutionary homological structure of the evolution. Next, few statements on earlier developmental stage of in a plant. Earlier developmental stage of plant. Here the correct answer is a PQR. The cells are flowers are deployed in nature. Cells of flowers are deployed in nature is one correct statement. Only some specialized cells in the reproduction organs undergone meiosis to produce haploid. Here the very important thing. So for the earlier stage of plant development, the reproductive organs undergoes meiosis to produce the haploids, and haploid cells are produced in a queue above, undergo the few normal mitotic cell division after the produce the haploid production. That after that haploid production only the cells are undergoing the normal mitotic process. So first is the meiosis, then it undergo the mitotic. These are all the correct statements. Okay, next question. Cells from an earlier frag blastula were removed from the animal pole and used to replace cells from a vegetal pole of blastula. Following event may expected. What is the correct statement? Here the question is, what is the correct statement? They are doing taken the cells from earlier frag blastula were removed from the animal poles. Okay, then it's used to replace the cells from the vegetal poles of the blastula. So this event expected. What expected one? So what expected means B is the answer. PQS. Okay, transplanted cells were developed normally as part of the cells of the vegetal pole. Okay, the next one, as per the statement here, replaced from the vegetal pole, right? So this statement explained that transplanted cells were developed normally as a part of cell of vegetal pole. And second option is what is the next expected statement is transplanted cells would develop as a cells of animal pole of the adult of vegetal pole. So it's a mixed one. That is here they mention the animal poles of adult on the vegetal pole. Next statement, remaining yes statement, remaining cells of animal poles would compensate for the cells that were removed. Okay, that are removed. Some cells were removed, right? That cells are compensate with the animal poles. These are all the statements which are relevant to this cell transfer from the blastula and replacement to the vegetal poles, that concept. So the cells from earlier stage of frag blastulas were removed from the animal poles. That animal that is the animal pole, that from that they are removed and that are replaced with the vegetal pole. So it's a mixing recombinations process. And one more thing about this this concept here, whenever they are given this kind of question, a mixed question, just read and understand the question. Here question itself they mention that is the some of group of animal poles are there, from that they are taken the particular portion and replaced with vegetable. So then what is the possibility? As the part of the vegetable poles is there, and mixed vegetable pole and adult pole, and its remaining cells of animals are composite with the removed cells. That is, the removed cells is extra cells. Okay, next, control of following is a complex process involving several key regulatory genes. Some statements on flower developments are given below. These are all the statements. Here, among this statement, which is the correct one? The PQR is the correct statement about the statement of flower development. Okay, the flower here, and this flower development, there are three proteins genes are there. Here they mention the importance. Two major type of gene regulates the floral development. Yesterday itself we are discussed the major type of gene regulate the floral development. This meristem identify gene and floral organ identity gene. A meristem identity gene and the floral identity gene. Important genes in Arabidopsis thaliana that play a key regulatory role in the meristem identity. Okay, these are all the meristem identity genes which are all in the Arabidopsis thaliana. That is the APETALA1 and LEAFY. Then suppressor of constant 1. Okay, these are all the genes in the Arabidopsis thaliana is a correct statement about the flower development. And the gene that determine floral organ identity will discover the floral hemotic mutants. Okay, this side, the floral organ identity genes are explained about the ABC model. Yesterday we discussed the ABC model, right? That ABC model is related to this floral hemotic model. So these three statements are right one, and okay, it's a wrong question here. The most plant homeotic gene belong to the class of related sequence of fat box gene. These fat box genes are not relevant to this floral development concept. Okay, so the we need to remove the this last option, and this fat box gene which having the it's expressing and it's promoting the binding of proteins in the sequence in the genes. Okay, it's present in many kind of plants. In the part of the plant, it's promoting the DNA photolase protein, the photolase process inhibition. Okay, whenever they are facing this fat box protein, this protein profiling is very complicated. In the latest researchers are focusing on the gene to understand their concept, because as per the till date many of the researchers are doing research on this FED, but they are not getting the better results on this FED concept. That FAD is nothing but that is a fatty acid desaturase enzyme. Okay, in this concept, it's normal in a normal plant system. They can able to face the various environmental condition, but that is not relevant to the floral development. Okay, here they may ask the floral development based thing. Next concept, amphibians, when due to some injury the eye lens is damaged. Okay, the fully differentiated iris cells can generate the lens. It's achieved through the following possible process. What are the possible process? Here you want to know one thing. In this coming questions are relevant to all the questions are relevant to research. Okay, they are asking the reason, they are asking the process. So we want to understand the basic research concepts in the theory. Okay, so there is a lens damage happen. That lens damage is overcome with iris cells. It's achieved by the process called the iris cells through some signaling undergone de-differentiation and trans-differentiation into lens cells to generate the lens. And next concept, the iris cells introduced in a stepwise manner specific genes responsibility of their differentiation and then conversion of lens cells. So first it's produced, then conversion now into the stem cells. These two steps are related to the this eye lens development process. And next concept about the in the presentation. Next question, a set of experiments that were carried out to demonstrate the effect of apical ectoderm rigid of the chick limb bud on the underlying mesenchyme are enlisted below along with their expected outcome. What is the expected outcome? The expected outcome is first one, removal of AER, that is the apical ectodermal rigid. Removal of the AER of forelimb leads to cessations of limb development. And if second option, if an extra AER is placed in a forelimb bud, duplicate of the distal region of wing take place. This is the second accepted one. Then finally the fourth option is the right one. In a AER, forelimb bud is replaced with a bead shock in a FGF2 gene. The normal wing is developed after this change. These three are the major expected outcome of this process. Okay, what process is the demonstration of effect of apical ectodermal rigid of a chick limb bud on underlying the mesenchymes are enlisted below. This mesenchymal expected outcomes are: first one is removal of AER, second one is extra AER placed in the forelimb, and the AER forelimb bud is replaced with the chalked FGF region. Next question, Henson node. Okay, it's a very important concept in the developmental biology. Henson node establish an avian equivalent of ambience dorsal blastophore limbs. This following observations are presumed to be supposed of the same. What is they are proposing? Hence node. As per their statement, the P is right. It's a region whose cells can both induce and the pattern of second embryonic axis when transplanted into other location of the gastrula. And another right answer, it's express the same marker gene as the Spaman organizer of the amphibians. These two reasons are there for establishment as a avian equivalent of amphibian trossel. These are all the reasons, two reasons. This region, it's a region those whose cells were both induced and the pattern second embryonic axis when transplanted into the other location of gastrula. Another answer is right. Okay. Another answer is the answer P. Answer R. That is express the same marker gene as Spaman's organizer of amphibians. Next thing about the question is experimental value. This in an experiment, the cells that would normally become a middle segment of Drosophila leg were removed from a leg forming area. Larva stage itself, they removed the leg forming area. So where plays a tip of fly antenna. So tip of fly antenna is placed on the particular space. Based on the French flag, analog for the operation of gradient of position information, which of following statement is true? They are asking the correct statement. As per their given statement, that first answer, the transplanted cells retain their committed status as leg cells but respond to the positional information of environment by becoming a leg tip cells. Though so two kind of cells are there. So they are facing the second or the first option A. So it's close. This is for example. Next question is, during the fertilization in a mammalian, sperm egg interaction is mediated by zona pellucida. It's a statement, a pellucida membrane protein that receptor present in the sperm membrane. ZP, that is zona pellucida 3, has been identified to be principal zona pellucida protein who whose post-translational modification is important for the sperm egg interaction. In a competitive incubation assay, the sperm is saturated with either active ZP, that is Xenophilicida, or its modified form before studying sperm egg interaction. Which of the following explains sperm's egg interaction? So they are given a big story, the story itself that having the answer. Okay, first step itself they mention during the fertilization of in a mammalian cell, the sperm egg interaction is modified by the zone of pellucida, that is the information, and the receptor present in the sperm membrane, that two things are interacted, zona pellucida membrane protein and the sperm membrane. Okay, membrane receptors are interacted, then it's identified to be principal ZP protein. Translation modification of that particular protein is important for sperm egg interaction. The competitive assay, the sperms is saturated with either active zone of pellucida or its modified form before studying the sperm egg interaction. So there was no study. Here they are asking the question, what is not inhibiting the sperm egg interaction? So they provided the possibility of interaction. Here they are asking non-possibility. Non-possibility is a deglycosylate the ZP, zone of pellucida 3 protein, and use it for saturation of sperms. This is not the reason for inhibition of the interaction between the membrane and egg interaction, sperm and egg interaction. Next one is human chronic gonadotrophin, that is HCG, is known to facilitate attachment of blastocytes to uterus. In a human with the mutation in a HCG gene, the biological inactive HCG was formed but implantation occurred. When HCG was immunotalized in the uterus of normal human, that implantation failed. So this suggested that implantation in a human, that is B is answer, blastocytes can produce the required HCG, which helps locally in the uterine attachment. This is the reason for that. Second one is during of reproduction development in the plant, what happened during of the reproduction in the plant? Option B, Q and S. Okay, the vegetative cells in the pollen contribute to pollen development. The pollen tube rupture and release both male gametes in one of the generated synergids. This already happened in the reproductive development in a plant. Flower development we are discussed in the earlier stages. Here this slide this explaining the reproductive development. First one is male and female gametes. First reason is vegetative cells in the pollen contribute in the pollen development. Second reason is the pollen tube rupture and release both the male and gametes in one of the degenerated synergids. This is the reason for the particular answer. Okay, next is very interesting, easiest question. And segmentation gene in a Drosophila are derived into three groups: gap, pair rule, and segment polarity. Based on the mutant phenotype, below are some of the major genes expressed in sequential manner with respect to the group affecting the segmentation part. Oh, this they are asking the which of the following the above observation sequence of gene expressed from earlier to late embryo is correct one? The correct one is D. Here the Q and S is correct one. Q, the hunchback, even skipped, and fusy trazus and wingless. That is the production stage. Next one is tailless, hairy, fuzzy trousers and gooseberries are produced. These two things are happen while that affecting the segmentation process. While affecting the segmentation process, these happen in the embryonic stage. Then polyspermy results when two or more sperms fertilize an egg. It usually lethal since it is results in a blastomerl with a different number and type of chromosomes. Many species therefore having two block to polyspermy, the fast block and slow block. In the case of CSIR genes, there is two possibilities are there. Which is two is correct one, means P and R is correct one. The fast blocks is immediate and cause the egg membrane resting potential to rise, which does not allow the sperm to fuse with the egg, and it's mediated by the flux of sodium ions. This is the reason. Another one reason is R. The first one is a fast block, second one is a slow block. Slow blocks are critical granular reaction is mediated by the calcium ions. So slow blocks are mediated by the calcium ions, fast blocks are mediated by the sodium ions. Okay, next is very interesting question and easiest question. Which of the following inference P and S problem and statement given below would you draw from the following issue transplantation experiment performed with the earlier and late gastrula stage? The stage is very important. In the earlier gastrula stage, prospective neurons and prospective epidermis are produced from the host region. The donor region are producing having the epidermis and then prospective epidermis and prospective neurons in the two stage. The differentiation in the donor tissues are the epidermis in the first one and the second one is the neuron. Then late stage that prospective neurons are produced and prospective epidermis are produced. The final output is neuron. And in prospective epidermis, the prospective neurons or epidermis are produced. Here these epidermis are produced in this stage. So as per their given statement, they are given statements here. That statements are explaining A is right one. That cells of earlier new T gastrula exhibit conditional development and cells of late gastrula exhibit the autonomous development. Okay, next interesting easiest question. Which of the following cellular communication source below will override the process of normal development? Sir, yesterday also we are seeing this kind of question, and here they are asking which makes a cancer. It's a normal which override the process of normal and lead to cancer. This is all the normal adult cells that having the lethal cell does not make a cancer. That here the delta signal and notch signal is activated. While notch signal is activated, there are tumor suppressor genes are activated, then that cause the cancer. This is a normal pathway. We are on apoptosis pathway, and caspase pathway is the apoptosis. RO, which is attachment adhesion pathway. Here they are asking the lead to cancer, which makes the cancer means that cancer cells to this movement, that is metastasis process is happen with the this process metastasis, and this is the apoptosis process. This is normal cell development. Adhesion is, and this is also the normal cell development. So cancer is it's a metastasis process, this is the apoptosis process. These two are making a lead to cancer. Okay, next question is the it's very easiest one, but the question is look like big. As per as I'm shared, this C-argin group of cells at the vegetal pole becomes specified as the large micromer cells. These cells are determined to become skeletogenic mesenchyme cells that will leave the blastula epithelium to ingress into the blastocells. This specification is controlled by the expression of PMR1 gene, which express the HESC gene. HESC express the gene encoding transcription factor activating cytoskeleton forming gene. So this HES activating the transcription factor and activating the cytoskeleton formation gene. And gene regulate network is given below. Mesomer and it's reset the cell blastula. These are all the sequential process. Here the below column 1 is listed experiments carried out with mRNA antisense RNA of different gene injected into the single celled CSIR gene embryo, while column 2 listed development occurred. What the process given here, this is the output, this is the injection of this chemicals that makes this outputs. Here they ask the question, what is the question means, the following combination is correct one. While you are adding the mRNA of PMR1 MAR1 to the CSIR gene embryo, that leads to the cell will start ingression into the blastocells. And then second one, mRNA of HES injected means that skeleton mesenchyme cells are not formed. Then antisense of PMR1 that leads to that stop the formation of mesenchymal cells, and if they are adding the antisense of HEC, that cells will start ingression into the blastocells. This very interesting, a very easiest question. While you are understanding the story, you can easily answer this question. Here this for just explanation, the maternal factors are two, OTX and the catenin, beta catenin, that activating the HEC, this HEC activating the this upcoming genes, this activation process. And this another one is the rest of cell blastula. They provided the confusion. This is the confusion state part up here. The question is big, but the statement itself having the answer for the question. Okay, fine. Today I was late, and I'm going to finish it now. Thank you for your patience, and thank you for your listening till the end. Now I am going to wind up this session. We will meet in the tomorrow class. I'm welcome to the day 17th class. Thank you everyone. I'm going to end this stream. If you have any queries, you can post it in the chat box. You can otherwise you can post it in Telegram group. Thank you so much.